I don't know if John is. Okay. There we go.
That just means we're recording.
Good. So have we got Dr. Farrell on board yet? Is he signed in? Okay.
I'm here, Peter. Yep.
Good. Okay, John, it's the session's all yours. We have a nice long time period for you of about 80 minutes to have, you know, both presentation of what you guys have been working on lately and to get some comments, questions, and concerns and observations into the mix for us as well. So, John, please introduce yourself. For those of us that for anybody that's here that doesn't know you, you are a phenomenal resource in the world of muskies, and we thank you for the work that you're doing. Over to you, John.
Thanks, Peter. I really appreciate the opportunity today, and wish to thank all the anglers for signing in this morning and participating, because that's really what it's all about. And it's just really exciting to see so many people with so much interest in the St. Lawrence River and the future of the Muskie Fishery. Can you guys see me? Because I can't see myself.
Yeah.
Okay, good. And I'm going to share a presentation here. So a little, let's see, it's just a little technical part of this. And there's our first slide.
Super.
There we go. Do you guys see the notes page or just the slide?
We see the part on the right that says next slide as well.
Yeah, we see the notes.
Okay.
Interesting. It doesn't really matter, but I didn't expect that. But
It's good.
no worries. There's just some notes in there. But so anyhow, I wanted to take a moment just to introduce a couple people that are, I think, out there. So we have John Paul Leblanc. So JP is an environmental scientist on our team at Tibbs, and he is a Canadian citizen and lives in Canada for half the year, and then he lives on the island the other half. JP worked in Patrick Fraser's lab, so he did a lot of amazing research on the Georgian Bay, and then after he graduated, we were fortunate enough to have him come join our group as a postdoc. He just recently applied for and received a position as a scientist at the Thousand Islands Biological Station. So JP, you want to sign on for a second and say hello?
Yeah, sure. Hey, everybody. How's it going? Yeah, I'm here. I'm going to be helping out. I have a little bit of muskie experience starting in Georgian Bay. Me and Dan Weller did a lot of telemetry and habitat assessment for muskie spawning in Nursery. And we looked a lot at water levels. And then I was fortunate enough to transfer my skills and ability to the upper St. Lawrence River, where we're still working with muskie and yeah, trying to fix a couple of noted problems. So it's a pleasure to be here.
Thank you.
Thanks, JP. And I just noticed on my title slide I got the date wrong. I do realize it's March 12th today. So, but just a little introduction on Tibbs. The Thousand Islands Biological Station was formed from a gift from the Lewis family to ESF back in the early 1970s, and it developed a research program that I've been associated with for a long time. I was fortunate enough to do my graduate work at ESF, but I did do graduate work at York University in Toronto. So I worked up in the Lake of Bays for a period of time with Don McQueen, and then came back to ESF. And so I got 2 degrees working on Muskellunge, and then my advisor retired, and I was able to stay and build the Thousand Islands Biological Station. It's just been a wonderful opportunity, wonderful community. And I learned from a mentor. So there's on Oneida Lake, there's the Cornell Field Station where I worked as an undergraduate. I just really enjoyed working at that field station, and saw how they built long-term database to understand population dynamics of important fish to Oneida Lake, like the walleye especially. And I recognized that the St. Lawrence really at that time didn't have a whole lot of that. The state agencies were definitely monitoring fishes, but we wanted to focus on nearshore fishes and fish reproduction. So we started some long-term databases that have really become valuable in tracking what's going on with the St. Lawrence River, and understanding all the ecological changes that I'm sure you guys notice and see, because so many of you spend so much time out on the river as well, that we, like Peter mentioned, we really value our collaborators and anglers that really feed us a lot of information and knowledge. It's kind of analogous to like indigenous communities where you get traditional ecological knowledge. The anglers are using the resources on the river. They spend an incredible amount of time out there. So it's incredible ecological knowledge to get from these intelligent people that are out just making observations as they fish. So we really value that. The Tibbs program, we have a little island. It's called Governor's Island. Stop by if you want. It's evolved into a pretty significant program. We cover a lot of things. Muskellunge, being the top predator in the system, have always been important. And I think our program is really founded over issues related to muskellunge conservation, dating back to the late 1970s, when populations were declining, and we didn't really understand things about like exploitation and their effects on populations and size limits. And so we were involved with a lot of that work. And there was a process that you'll hear about. It's formed under the Great Lakes Commission. So there's a Great Lakes Fisheries Commission that kind of oversees the fisheries of the Great Lakes. And there's a subcommittee for Lake Ontario. And under that, there was a working group established to deal with the issues related with muskellunge. And there was members from the US and Canada that were formed. And I was a graduate student at the time, but I got to work with some, remarkable scientists. Ed Crossman came out to the island several times and worked with us, and John Castleman and John Forney, who I mentioned from the US side. These people were just my idols. They were incredible gentlemen and fisheries scientists that I learned so much from. So the working group is still in existence, and it formed this legacy of informed muskie management, and working with anglers is a big part of that. So today we're gonna talk about some of the issues we're seeing with the production of young muskellunge on our nursery grounds, some of the concerns, and we're trying to help rebuild that. And we're launching a new kind of muskie citizen science program. And I want to thank Rob McRae and Peter and all the others that have been involved thus far. So this is great to have this opportunity to let you know a little more about it. Okay. So this is kind of the NASCAR photo. We can't do anything without partnerships. So there's so many partners, and I see a bunch of names on the list here today that have helped conservation on the river so much. I'm not going to go through each one, but I just want to mention our agencies. New York State DC. This is that's funded us since 1987. We've had consistent funding thanks to New York State DC. There's a name I want to mention, which is Steve LePan. They say Steve LePan is the original muskie man from the scientific standpoint, but he helped form the project in the early days, along with my advisor, Dr. Bob Werner, who recently passed away. So Steve just retired, but he really, put his heart and soul into making sure this project received adequate funding for us to do our work. And we also learned so much from him. DEC has funded the program, and then there's just so many other important partners here. Save the River is another one. They started the Muskie release program in Clayton. That's been, pivotal to helping, provide an incentive, and the attitudes of anglers have changed. And then the Thousand Island Land Trust has been doing land conservation, and they've actually been protecting a lot of muskie habitat on the U.S. side. So it's they have a significant land holding in Flynn Bay, which is one of our main spawning and nursery sites that's protected in perpetuity. So we're really excited to have all these elements: Save the River with advocacy, the land trust with land protection, and then the angling groups involved, because they're really the eyes and ears on the water and handling the fish, and then the agencies and scientists to provide all the information to track populations. It's so I guess the point is that all the elements, at least on our side, seem to be in place. And I see the same kind of things on the Canadian side as well. So we have Canadian partners. We've been working more and more with Colin Lake. There's exciting news from the ministry that they're going to be launching a program to start really focusing on Muskellunge. And I think you'll hear from Colin after my talk today more about that. So we're really excited. That's a big piece of news. And then we also work with Matt Windle, and Matt is at the St. Lawrence Institute in Cornwall. So seining is going on and looking at young of the year production on both sides. And I know the GAN chapter has also done a lot of inventory work, trying to identify these important critical reproductive areas so we can try to protect those and make sure they can continue to support the population. So that's just, I'm leaving a lot of people out, and I apologize for that, but there's just so many partners involved with this that I don't want to take the whole day to talk about them, but they'll probably come up. Just to review the management. So there was 3 management plans have been written. They've kind of evolved. We're in the process of developing a new management plan in concert with Ontario, and we're going to certainly have opportunities for anglers and angling groups to participate in that. It's just in its beginning, but there's some core goals that I doubt will ever change. That the one is that the goal of having a self-sustaining population of Muskellunge is just really critical. I view the St. Lawrence as a self-sustaining population. We'll show you some data that this is kind of an aspect that we have some concern about. And second is the logical role as the apex predator. So they do serve a really important role. You really kind of need to know more about in the St. Lawrence River fish and aquatic community. They're top dog. I have examples of catching adult muskie, and I think when they come into a trap net first, I've had cases where it's the only fish I'll catch in the net. So I think once the other fish know muskies are around, so there's kind of a landscape of fear, if you will, that's important to the ecology, because they can pretty much eat anything they want out there. So it's really important to have that apex predator in the system. And then the other goal is the recreational fishery. So the quality trophy fishery that you guys use and help maintain. So it's just three goals there. And then I mentioned the working group. Back in the day, there was very little biological data and monitoring. That's changed. we know a lot about the critical habitat, but there's such a complex ecosystem there that there's a lot more to know. So we continue to identify critical habitat to try to protect it. I think the protect part is the part that's really been under fire. We've just, as you guys know, we've had 2 major floods in 2017 and 2019, and it's led to a lot of shoreline hardening. There's been a lot of money invested into rebuilding, and I totally understand that, but there's also, you know, a lot of development, and the siting of development and the protection and making sure that the permitting agents and the community understands where these critical habitats are is going to help us protect them, because, you know, I don't think the regulatory framework can do the whole thing. So we're really looking at, you know, protecting our habitat. I don't know if people are aware, there's a recent controversy because the Customs and Border Patrol are right now proposing a development. And I'll mention a little bit more about this, but it's a development at Fisher's Landing, which is between Clayton and Alexandria Bay, really just upstream from the Thousand Islands Bridge a little bit. There's a large embayment, and they're in the interior of the embayment. So they're proposing a docking facility, and there's been a big outcry about this proposal, but it happens to fall right on top of one of the prime muskie habitats on the U.S. mainland, and it's at risk of being altered. So there's a lot of need to protect our critical habitats and then improve fishery management. So, you know, hopefully we can have some discussions about the fishery. You know, there's one aspect is the muskie anglers themselves and how they do their business and pursuing this amazing fish, but there's also, you know, other fisheries that intercept and affect muskie populations. So hopefully we can have some discussions on that. This is one of the few management plans that's actually been evaluated. So I've got a paper here, and you can see John. So there's U.S. and Canadian participation. This is a paper from about 10, 12 years ago that evaluated the management and how the population is changed. So, you know, we're kind of working hard to do this, but it does seem to be one of the better managed species in the Great Lakes, and it's really been like externalities that have created some difficulties. So the management plan, again, here they have a goal. So this is the original goal hasn't changed, to perpetuate the muskie as a viable self-sustaining component of the fish community in the St. Lawrence River and provide a quality trophy fishery. So, you know, we've seen, like in many musky waters, we've on top of the critical habitat identification, we've seen increases in size limit, but it's really been this voluntary catch and release that has had a major impact on the quality of the population and fishery. So the river's changing incredibly around the Muskellunge, and that's where a lot of the research and science comes in, is trying to understand like how all these changes are in fact affecting this really quite. It's remarkable. It's such a large voracious predator, but it's also a sensitive species. So in New York State, Muskellunge is actually listed as a species of concern. And at the same time, you know, we wanted to make sure that the fishery remained open. So the working group, you know, allowed that to happen, which, you know, is because of the declines I'm going to talk about that we've seen in our nursery grounds. Okay, so the research program, you know, we do adult population studies and monitoring, and the angling community is part of that. There's an angler diary program, but we also have a spring spawning assessment that we do, and we follow that up with seining series, and we try to do this in a standardized way, so we can look at what's called a longitudinal study. So that's a study that occurs in the same way over time, where we do monitoring. We can look at population changes. We've done a lot of work on spawning habitat, actually mapping egg distribution of spawning muskellunge and computing survival. We've done a lot of work with young of the year habitat, distribution of young of the year in the river, and also with insights and survival of young of the year. So these are just decades of studies. We've done work on these subadult stages. So, there was kind of a gap in understanding for these really young muskellunge after they leave these wetland nursery grounds. And we've done a couple of studies, recently worked with Steve Cook's lab at Carleton and did a joint project between US and Canada, which was really cool, doing with a acoustic telemetry study on age 1 fish. And we do a lot of habitat studies as well. We monitor the forage base. So this is kind of interesting because, you think of like that other fishery, the Lake Ontario salmonid trout and salmon fisheries, it's all about like understanding the forage base and looking at the predator and prey relationships and making sure they're in balance. These concepts really haven't been applied to muskellunge, but the same concepts occur in the nursery grounds. So the forage base that exists in the nursery, looking at these, like what's called fusiform, which is kind of torpedo-shaped prey of the right size. So looking at the minnow community and things like killifish and other species that are really crucial to the muskellunge as a forage base have been altered by round goby. So there's some really interesting research going on in there. And fish community monitoring and research. So we can't look at muskie in a vacuum. We kind of look at a whole array of species that muskie depend on. A lot of work on population genetics. So we've had partnerships with Louis Bernacci out at Laval University, has done some really amazing work, and also some people in Wisconsin, Lauren Miller and Chris Wilson as well. There at the Trent University lab. So a lot of work, and this is where anglers really helped us. We collect genetic samples, but in the past we've had muskie anglers in our diary program collect small tissue samples, just a tiny little clip of fin that goes into a jar of denatured ethanol, and we can do population genetics. And there's some really good news there, because there's been a lot of stalking from outside sources in the system over the years, principally from Chautauqua Lake. And what the take-home message from the studies is that the main stem of the St. Lawrence seems to be pretty much immune from that. So you can get what's called integration of other muskie genes. They're evolved to really perform in the environments that they exist in. So when you take like lacustrine or lake forms that are from the Ohio River Basin, they have very different sets of traits. And if you mix those with the St. Lawrence, the concern is that it would dilute the native population genetics. But what we find is that a lot of that stocking seems to have had no influence on the main stem muskellunge. It's interesting when we look at populations that are adjacent to the main river, that's where we see influence. So it's important for us to maintain our regional strains. Disease dynamics. So we work with Cornell University. John Lunston at Guelph was the one who first identified the VHSV in the outbreak we're going to talk about. That's viral hemorrhagic septicemia, which has had such an impact on muskellunge. The virus is still there. We're studying it with Cornell Veterinary College. And I have a PhD student, Anna Conklin is her name, and she's really focused on the disease. It's amazing after studying this for a period of time how analogous it is to like COVID. So we see there's probably over 100 variants of VHSV now in the fish. We know that goby are a really important reservoir for the virus. So it's still out there, and it's something that we have to continue to track and monitor. And then population viability and restoration is another area we're looking at now. So I'm going to really be focusing on that, trying to maintain healthy stocks to provide those services that we talked about. So critical habitats, we've early on used radio telemetry studies with these older technologies. to identify sites. And then there was a lot of seining that occurred. And there's just a wide variety of habitats that have been identified in the system that are currently known to have produced juvenile muskellunge. So these are the ones that we really want to make sure that these habitats stay viable. And that's why so much effort goes into this, because it's really the backbone of the population and fishery. And so much work has gone into this. So if you guys have questions about it, we can come back to it. So I mentioned viral hemorrhagic septicemia. So this virus came in. I think the population was really heading towards a new all-time high when it hit. And VHSV is a rhabdovirus. This is a picture of it here. And there was a 3-year period where John Castleman and I and others, muskie anglers, tour boat captains, we started seeing these large adults floating all over. We picked them up. I picked up this 59.5 incher you can see on ice here in this tank one morning when we were out trap netting. It had a couple of sores on it, but we think that was a different disease. It's not like an external thing you can see, except for, it causes internal hemorrhaging, and it really is lethal to muskellunge. There's been a lot of research done, and VHS can infect like a lot of different fish species, but there's some that are incredibly sensitive to it, and that was the case with muskellunge. So there's a lot of research on, and we're seeing this high prevalence in Obi. It continues to be an important reservoir that maintains the virus in the ecosystem. So, we're kind of worried about the legacy of the HSV. So here we have an angler diary program that goes on, and I thank all our participants. It's kind of a small program, and we hand out these diary books. There's now chapter 69 has created a electronic version of the diary program. So now you can log on and actually use the internet to load in your diary data, but some people still prefer the books. So we've been monitoring the fishery since 1997, which is part of the management plan guidelines. And, we've seen this, the effort goes up and down, and this should be viewed as kind of a biased sample, because there's like different anglers will come in and participate. So we try to keep like a critical mass of anglers, but they've changed over time. There's only a couple people that have fished this entire period, and you can see these fluctuations in effort. But when you put a regression line through it, it's really flat, which is I like to see. So our diary program stayed around just below 600 angler hours. So this is just a sampling of effort on the U.S. side. And their catch rates have kind of fluctuated as well. You can see these relationships between the catch and the effort, and there's a high correlation until you get to about 2012, they start to deviate. So effort is going up, but catch rate goes down. And then we have, some interesting things that happen. There not that goes into diary program. We have their expert, I think, that's on the call. That will be talking about the lunge log is the other big program that goes on that's so much larger. But we've seen this kind of slide in the success of the anglers. This is how we manage the fishery. So the goal is to stay at about a catch rate of 0.1 fish per hour. So every 10 hours fished, you would catch, it used to be a legal sized muskellunge, but that was established when they were the size limit was much lower. So it's now a muskellunge, because you would never catch a 54-inch muskellunge in 10 hours of fishing unless you got really lucky and stopped. So it's kind of like going to the casino, right? So the only way to win is to quit. But the star here is this really encouraging. So that, as you guys know, this is your data. The fishing was pretty good last fall, and we're seeing that. We're still getting in our data from last fall's fishery, but right now we have 472 hours and they caught 34 muskellunge. So that would give us a rate of 0.072, which is one of the best rates we've seen in a long time. So the catch rate was really good last fall for 2021. So it's going to spike back up and the effort's down. So it'll probably look a lot like this 2018 point where the catch is up and the effort was down. But we see this long-term kind of slide. So that's something to discuss. It's a little bit concerning, but the more concerning thing, let's see. Oh, that's not supposed to be there. Is some of the other data we're seeing. So I mentioned that we do an annual trap net survey. So we go into our nursery areas and we set a couple different types of nets. This is a hoop net. You can tell this is set in low water, just kind of so you can see it, but it has a series of boats. There's a lure that goes to shore, and the muskies are spawning, and there's better spawners, so they encounter that lead and instinctively go deep. It's like a minnow trap. They go through a bunch of throats. They end up in the end. We check these things daily for 30 days from May 10 to about June 10, and catch the spawning runs. Some of the warmer sites we start a little earlier, but the river is pretty sluggish to warm. So you can see that leader. This is an Oneida trap, which has a 6x6 box. So it's a different type of trap. So this is the kind of sampling and one effort that helps us track the spawning population. And these are net locations that we've sampled in the last 5-6 years. Let's see, I want to go right to this slide. This is kind of what the data is showing. So when we look at this long-term data series, we can kind of see population trends, and the catch rates were really much higher. This is actually the numbers of muskellunge caught in our survey by these different time periods, and you can see this slide and the number of spawners that we're seeing. And we're indexing like 11 sites throughout the entire 1,000 islands on the US side. We also have an exploratory series that we compare to this data. So there's like an index and an exploratory. And we've increased effort. So we've added nets to be able to handle muskellunge. So at the same time we're adding nets, we're seeing a reduction in catch. So we're pretty concerned about this trend. And it seems to be separated by the VHSV event. And then the thing that we're really concerned about is we also do this seining survey. We've been doing this a long time. And we're looking for the young of the year muskellunge. You can see a picture of one. This is kind of what they would look like, in maybe midsummer, early summer. About the size. And this is our catch. We do 2 surveys. There's one with this smaller fine mesh seine that we do, and we start July 15 and go to July 31. And then there's a larger seine that we use when they get bigger. It's twice the size in August 15 to August 31. We do that survey. And, we've been tracking, this is some data since it's been standardized. There's a lot of earlier data that actually goes back to the 1980s as well. But we really standardized the program to 11 sites that we sample throughout the region. And each one of these data points represents 90 seine hauls, okay? So a lot of effort goes in to catch the muskellunge and index their reproductive success each year at the nursery grounds. So there's 90 seine hauls per dot. So you think of the amount of work that went into maintaining this data set. It's one of our most important and valued data sets. And we also collect information on the habitat of muskellunge, all the other species. So there's probably like 50, 60 fish species that show up that we track that are important to muskellunge, and we collect other data on them. Like we'll take a little tiny thin clip and get genetics. And you look at this period of when we saw those mortalities, there was a regime change. So we used to have really, really good catch rates of muskellunge in our surveys. And then after that VHSV die-off period from 2005 to 2008, things really tanked. And then they tanked again more recently. I purposely left out 2021 because I'm going to show you something, but we had 2 years, 2019 and 2020, the catches were zero. And it's been quite remarkable to go from abundance of these young muskellunge all over our nursery grounds. We were so optimistic at this point. We're seeing, so you see these really high spikes. Those could be strong year classes of muskellunge. So, you know, look out like in 10 to 15 years from one of these, because it really influences the fishery, because this is what it's based on. So that's why we're so concerned before and after this VHSV period. We've seen this decline. And you know, Matt Windle on the Canadian side, and also the GAN chapter and all the other seining work that's been done. We also do exploratory seining. They collaborate what we're seeing here in our index. So at first it's like, is this index correct? Is it accurate? Has there been a change? We've tested those ideas, and we really don't see a lot of muskie on the nursery grounds like we did in the early period. So this is why we're so concerned. And that's why we started to think about taking some action to see if we could, you know, change that trend. So we began this population enhancement study. There was a couple important points. So when I did my PhD work, I mentioned that survival stuff. So I actually stocked fry, these little 1-inch fry in the river to study, not to restore the population really, but to study muskellunge. So I released fry in like 10 sites for a period of 6 years and developed survival estimates. So we have a baseline, and we know, I did actually some fingerling work too. We have baseline estimates before all these changes in the habitat. So we know how these things should perform relative to when I did my work from 1990 to 1996. So we have all this information of how these fish should perform. And then we did a little pilot study in 2013 to 2015, where in 4 sites we released these fry and did a little pilot study. And then more recently, we're doing a full-fledged assessment of a population enhancement study. So that's what I'm going to talk about. So we take adult muskellunge. We very carefully strip eggs and fertilize them in our lab at Tibbs. And we incubate them to the fry stage. So we have a fry stocking program, and then we also incubate them to fingerling stage. So we're kind of comparing these two methods and their performance. And I'll tell you a little bit more. We've been partnering with U.S. Fish and Wildlife Service. So this is Scott Schluter. He's incredibly dedicated to the river. He works on sturgeon, walleye, and also on the Muskie project out of the Cortland Ecological Services office, and he is a partner. So they are running nets down in the Lake St. Lawrence region during the weekdays. We cover them on weekends, and then we do the Thousand Islands. Okay, so I mentioned the release of fry and fingerlings. We're focusing on restoration of lost spawning in nursery sites. We use local broodstock and genetic strains. So there's a genetic protocol we have, and then we're doing a lot of research and monitoring on the performance, growth and diet and survival of these fish as they grow and develop. And here's more on it. So these over here on the upper left, these are early life stages. So that's a muskie egg that's been fertilized. You can see the backbone. So it's amazing, these fish that reach the proportions of what you guys catch start out is saying about the size of a Crossman BB. And they lay like in their what we call demersals. So they lay on the bottom of the river in these wetlands, and they're like so vulnerable to like temperature shifts, oxygen changes, predation by round goby. We've studied a lot of round goby predation on eggs. And they eat them. Other species probably eat them. So there's so many challenges that they have to go through, that their natural survival rates are pretty low. This is a sac fry, so they hatch out in a couple of weeks, depending on temperature. And you can see the one on top is a Muskellunge. They're beautiful. They got this gold yolk sac that they absorb as food. This dusky one below it, that's actually a pike. So it's comparing a pike and musky sac fry. So you can, I'm teaching you guys, you can identify the two by the pigmentation on the yolk sac. So if you ever see one, but they're about 1/2 inch long. And then once they absorb that yolk, they'll take a gulp of air from the surface of the river and inflate their swim bladder, and then they'll start to feed on zooplankton. So for a couple of weeks, they'll feed on zooplankton, and we feed them Artemia, which is, if you remember sea monkeys as a kid. So we culture these sea monkeys in mass and feed the muskies those things, and they just chomp them up like popcorn, and they grow. And we mark them as a batch mark. I'll get to that. And at Tibbs, we have all these raceways to raise these fish. I invite people to come out and check it out while we're doing this. It's a pretty exciting time. It's a lot of hard work. So we raise these things to an advanced fry stage. So these advanced fry are ready to shift to feeding on fish, like really tiny larval fish. That's what they need. They don't eat insects and stuff like that, like other species will. They go from zooplankton and they start feeding on tiny fish. So there's something called a critical period where they need to find these forage items in order to survive. And then we let those go. We'll talk about that in a minute. And then we have another system where we retain a bunch of them and start feeding them this BioVita dry pellet fish diet. And then we'll shift them to minnows, which are really expensive. So we minnows and then shift them to minnows. And we do intense rearing in our tank. So you have to really route them to get them excited to convert to the dry diet. So we've learned a lot of the techniques from hatchery people and also through experience. So you got scientists raising fish on Governor's Island, which is it's been fun, and you learn a lot by raising them. They're very amazing. We end up having to cover the tanks because they're so wild that they jump out. They'll jump out of coolers when we go to stock them. You just look at them the wrong way, and they'll, we're afraid they'll dart into something and break their snouts or something like that. So they're incredibly voracious. And we do a post-stocking evaluation. So these little fry, I mentioned the batch mark. So we immerse them in this chemical, which is kind of like an agricultural antibiotic, but it's called oxytetracycline, and it leaves, this is the otolith bone. So every muskie has an otolith in its inner ear, semicircular canals, and the otoliths lay down a daily ring, just like a tree. And we actually can detect that day that it was immersed in this chemical by dissecting out the otolith on a subsample of these fish and then transmitting ultraviolet light through it with a special microscope, and it'll actually glow like that. So we know it came from our lab. The larger fish are given a mini pit tag. And this is where the anglers come in. We're really in need of identifying these tags, because they're going to be, the plan is that these are permanent in the Muskellunge, and we can find them when they grow to adulthood. So they're injected just before the on the back of the Muskellunge, on the dorsal fin. So there's there we are releasing fish. Here we are doing the assessment. So Tibbs is now doing this near-term monitoring, the first season of growth of the Muskellunge, and then Tibbs and the ministry are going to be engaging in this spring adult survey. So we're really excited. We're going to hear from Colin about the adult survey that the Canadian agency is planning on doing a little bit later. And then we're hoping anglers are going to assist us with the monitoring of the older age classes, the adults, and looking for these tags. So that's one part of the citizen science program that we're discussing. So we've been doing some stocking. These are fingerling stockings that have been done in the river, and they're designed to restore a bunch of these spawning sites that have really lost a lot of their vitality, and that's been determined from the seining program. So this is 2017, the 2 years of the floods. We were pretty successful in getting fish out in the river. We actually stocked 4,500 fingerlings in 2017, about 540 in 2019, and all these fish were pit tagged. So these fish are going to be 5 years old and start to enter the fishery, we hope, this fall. And these are the different sites and the numbers. So the cool thing is we know these individual tags have an alphanumeric code that links to that individual. So we know where it was stocked, when it was stocked, how big it was stocked at. And then there's all sorts of questions we can learn about where these things are recaptured. We'll finally be able to know if they home to the same sites exactly. There's a lot of evidence for homing in Muskellunge. So there's a lot of interesting biological questions that can be answered alongside trying to help restore spawning and nursery sites. This is from 2021. We were successful in 2021 and collected eggs. You can see our rearing jars full of muskie eggs. These are the batches and the pit eggs that were used. These are the fertilization rates that we saw and the volume of larvae. These are larvae here, and here's an individual that's blown up. So this fish is about 3/4 of an inch. They start to look like little muskies, and they're feeding on the Artemia at that time. We stocked a much broader set of sites with advanced fry. Over 38,000 advanced fry were stocked across 46 locations, and we do it at 750 fry per hectare of habitat. And they were done from Eastern Lake Ontario all the way to almost Waddington. So we covered most of the important spawning habitat. And this is the distribution of fry. And then we've been evaluating their success. Last summer, we spent a lot of time seining and evaluating their success. This is some of the growth data from the fish. So what was exciting is that we saw muskie on 54% of the site visits. A little different than the zeros we've been getting. So we were very excited about how we were able to increase the densities of young muskellunge on the nursery grounds. So we saw these increased catch rates. We also saw some natural reproduction. We had recaptures of the pit tagged individuals, which were exciting to see them, some of them multiple times. And this is the growth distribution. So they were putting on weight. So these fish are getting up, around 7, 8, and even one really large individual there. So pretty big size. So they went from fry to looking like this on the nursery grounds. This is a catch from Thurso Bay, which is right across from Gann. We haven't seen catches of muskies like that in seining in many, many years. And you can see they're stocked at this length, nice growth, finding forage. They're growing. This is what they're looking like in August. And when we add 2021 to the seining distribution and that sad story I was telling about how they've declined, when we look at this survey, we see a huge jump in the catch per unit effort at our sites, and it starts to look, a little bit more like what we've seen in the past. So that's kind of good news that we were able to put muskellunge back on the nursery ground with these fry, and we didn't even have fingerling stocking that year, but the fry did really well. We could talk about why we're using fry, but the fact that the habitat can support those early life stages and produce nice fingerlings on our nursery grounds is a good sign for the possibility of recovery of some of these populations. So what's out there for tags to detect? So the angler program, we have tags in adult muskellunge from the spring spawning. There's 97 tagged fish out there right now. There's probably been some mortality, but that's what we've tagged, natural mortality. And then through fingerling stocking, there's 5,000 fish. And we're continuing to raise and put these fish out there. And now with the new adult programs, there's going to be more tags to detect in the system. So we're hoping that people will be fishing and be able to, it's going to be really exciting, the first tag that's detected that came from this project. So taking a little shift to the citizen science program. So there's kind of a workflow here. So we talked about the hatchery and the tagging of the fish, working with the agencies. And the question now is it really having an impact on the fishery and the population of adults? So we need help. What we're proposing is that the anglers will get a kit that they can keep on their boat that would have one of these pit tag wands. And I want to have a call out to a couple people, Rob McRae and also Trevor Thiem of Chapter 70 on the US side have been purchasing through, and Muskie's Inc. and Muskie's Canada have been supporting the purchase of pit tag readers. So I think we have 5 or 6 readers that are dedicated to this program now. And we just put in several grant proposals to produce these kits, and our goal for 2022 is to have 10 of these kits on each side of the river. So the Canadian anglers would have 10, the US anglers would have 10, and they would be trained in collecting biological data on the muskellunge they catch. We hope to minimize like any negative impact to the fish, but also negative impact to your fishing experience when you're out there. So, trying to minimize handling and how much time this takes. So we're proposing some trainings that we're going to have, and it would be fun to get together and talk about this. But and then that data will go back to TIBS, and we'll be able to reference these tags in our database. So we have a large database that kind of tracks all these numbers and where the fish come from, and hopefully we'll have some exciting stories to talk about. But that's kind of the flow of ideas here. So the kits being provided to citizen science anglers would have a pit tag wand. These things cost about $850 a piece, just the wand. They come from a company called Biomark. They work really well. We'll talk about these pit tags and injectors. So this is the Biomark injector, that's the tag. So we want to read tags, but we're also talking about the possibility of having anglers apply tags, which we'll mention a little bit in a moment, because there's some complicating factors with that. A measuring tape would be in there to get the length of the muskellunge. And then genetic tissue samples. So there'd be a vial filled with ethanol, and then some scale cards to fill out the information, piece of scissors to just get a tiny fin clip off the dorsal fin. And I think Colin has some information on that as well. And we're going to supply these 10 kits to US and Canadian anglers if they're interested in helping us in this endeavor. We're proposing some workshops. So right now, the Antique Boat Museum in Clayton has volunteered their facilities to host a workshop, and we're looking at one in July and August. And we're also hopefully, we're going to be able to cross the border freely. We'll see what happens. But we could also host some workshops in GAN or other parts of Canada. So we're looking at doing a few workshops. ESF has a video production unit, so we're going to videotape the workshop, and especially like the handling and the biological data collection. So we would have that for additional training in the future, and also just to have a reference. So there'll be a nice video made. So the legality of this. So to be able to do this, we need to have participants listed on this thing called an IUCOOK. An IUCOOK is our animal use and care procedures. So we have to submit this large document to be able to work with Muskellunge, and it would list all the participants, and then they would have that on the boat in case they're pulled over. They could say that they're part of a research study. and it would be on our side. We're evaluated by veterinarians, and there's all these regulations that allow us to work with vertebrate species that we have to maintain. So the plan is to have participants listed on our permits. And Colin's going to refer to this maybe more, but in Ontario, there's a separate permitting process that would be required, especially for insertion of tags. And I hope today we can discuss about what we hope to learn together, because there's a lot of exciting possibilities. So that's a discussion thing. So in summary, I think we're in the second time to help restore the muskie population. We've been through this before. It took citizens in a community, like Peter mentioned, to make it work the first time. Natural reproduction equals sustainability, and it makes this fishery and population so special. We've got all this management and partners in place already working together. We seem to get along. Brian was scaring me a little bit when he was reading out the rules earlier, but I really feel like we have a common goal to really protect this amazing species in the system. We have an adaptive process through our management plan, and that's being updated. The wild card is this, invasive species, and VHS have created uncertainty and has like the loss of and change of our critical habitat. So, we know the river's changing, and we're trying to help this native species be able to be sustainable for future generations to enjoy and to enhance not just the quality of our fishery, but the quality of the environment in the river. It's going to take collaborations, commitment, and funding, and everybody's already piled on with this. And you just see it happening with this group here today. Protection, restoration, and management are key themes to protect the Muskellunge. So that's what I have for my presentation. There's a little how you can help thing here and here. We started this Friends program for TIBS, and people are contributing funds. We're in our first year to help support the field station and the Muskie recovery program. There's also options to participate in our angler diary program. Dan Talon is, I think, here too, and he kind of manages diary programs on the Canadian side. So Dan and I collaborate on that, and we need to do so more. And then, support these organizations, which you're already doing. So these are, broad messages of people can help. And the fact that you're here today is really hopeful. So I'd like to end with that and open it up to questions and comments. We got JP. I heard Arunas is here. I don't know if Arunas is on the line. I think Matt Wendell's here. So you got a bunch of experts, and hopefully we can learn from you and answer some questions. So thank you for listening to my talk, and I hope you enjoyed it. And I'm turning it back over to you, Peter, and I'm happy to answer questions.
Thank you very much, John. What a interesting and right up to the minute presentation. There's some encouragement there, but I think, from the point of view of the Muskie community, we're really glad to be able to work together to be able to help with the situation. The citizen science approach, I think, has always been core to Muskies Inc. and to Muskies Canada in helping out with reporting and angler diary information. But we've got a good broad cross section of people that are in on the call. There are guides, there are recreational anglers, and as you mentioned, there are some fisheries management experts and whatever. So why don't we open up? Are there some questions out there? Who'd like to be the first on the dance floor?
Just add as well, if you want to put a question into the chat area, you can do that as well. You don't have to jump on a mic. You throw it in the chat, we'll pick it up.
I'll throw a first question out there. First, just to thank you for all that you've done and all that you do, Dr. Farrell. And fantastic to be here. I wonder if you can put a price per fish for the fingerling stalking program and for the fry stalking program. I know your hatchery is an incredible resource and it's costly. Just so we get an idea on the Canadian side, how much does it cost per fish that went in the water? Fingerlings and fry.
Sure. Thanks, John, for your participation and the question. And it's probably one I can't answer very well because, you know, we're not a hatchery. I know there's data from hatcheries. You know, I say we're not a hatchery, so it's kind of like experimental, right? So we built this facility ourselves, and it's not like the Chautauqua hatchery or something like that. But you know, and I've seen some published numbers of what it takes, and then, you know, and being involved with the culture, when you add like the time of all the technicians, and like JP and I like spend incredible amounts of time raising the fish, and I really don't keep track. I don't want to keep track of it because we're in there like 7 days a week. We started you know, 7 a.m. and we shut the lights off at 10 p.m. and we live on the island. So it's like, you know, they become like your children kind of. I'm sure JP's smirking because he's worse than I am. We'll like have a competition of who's going to be down at the lab first in the morning. And then, you know, you're trying to run everything else. So I know I haven't really computed the cost, but it's substantial. And that's numbers of like $10 to $50 Muskellunge, fingerling, and stuff like that from professional hatcheries. But I could get back to you on that, but I don't know. But the cost is substantial in our time, because, you know, we're managing like so many other things, like all the science and all the other obligations and projects that Tibbs is doing. And then we have to like, so we do have a couple grants. I wanted to mention the Fish Enhancement Research and Mitigation Fund, which was formed from the relicensing of the power dam. So Scott Schluter manages a fund that was originally, I think, about $24 million. That was part of the settlement of the big dam. And it was designated towards fish conservation, habitat conservation, and they've done so much with it. And we are funded by them and then also by the state. So I know, you know, we just do a lot of work and we get we have a budget and it fits in there. And I'd have to do the calculations on cost per fish, but I bet it's like 10 to 20 bucks a fish, you know, but the fry would be much cheaper. So that, you know, the appeal of the fry is that it's quite a bit easier because the feeding regimen is so different. So you get the eggs, which is really difficult. You know, we sample 44 sites for a month to be able to handle enough muskellunge to get the eggs and do that. And then we incubate those. You know, they go out before July 1st. So they're out of our lab, out of our hair, into the system at 1 inch long. And you just have to raise them with the brine shrimp. It's a lot of work, but I think it's cheaper than the fingerlings, where you have to train them to feed, and then you keep them in the lab, you know, till well into August. So it's a lot more effort feeding them minnows. Disease is like the thing you have to worry about. You have a disease outbreak, the new treatments, but so I hope I haven't really answered your question, but it's costly. Thanks.
John, I wonder if I can ask a question. In the observations that you pointed out related to kind of the natural reproduction and how challenging that seems to have been, you know, is that situation really happening kind of, from what we can see, uniformly on both sides of the river? I mean, there's a lot of documentation that you've been doing over many years on the New York side, but can we really expect that it's pretty much a common situation that's happening also on the Ontario side? I think Matt might be on the call. I know Rob McRae's on the call. We can maybe look at that. But, you know, this is, I guess what I want to get at is whether or not this is a New York phenomenon or it really is part of the whole ecosystem.
Yeah, that's a good question. We've thought about that, and you know, that I don't think the ecosystem recognizes the political international boundary in the river. We're hopeful that, though, that, you know, because of the geologic variability in the river. You know, we've got like the Frontenac Arch that goes right through the center and cuts across, and it has that A2A corridor. So Algonquin to Adirondack corridor creates kind of different habitat. And then we have like the limestone on each side of that. And, you know, we kind of know what good habitat is and what it's performed like in the past. And there was a biologist, Ann Bendick, that worked for the ministry way back. And she, like in the old days, she identified a lot of spawning habitat. And then I know the GAN Muskie Canada group really got active in identifying habitat. They really rose to the call and did a lot of screening and got trained in it. And then, you know, we're really blessed to have Matt Windle join in and start his fins program on the Canadian side to learn not just about muskellunge, but about fish communities on the Canadian side. And you know, we did the exploratory sites. So we seine many sites from Eastern Lake Ontario to Lake St. Lawrence pretty much every year now. And so we feel we have the index really provides a good handle. And I don't think anything that has been seen in the Canadian sampling would really like change the view. And Matt's been developing some sort of an index, but I think his sampling is not targeting muskie habitat specifically. They do, and I'll let Matt talk about it. But you know, I believe that you know, the catch rates kind of speak for themselves and represent the St. Lawrence, and we're going to get more information because you know, like this past year, our catch rates were incredible with following the fry stocking, and I don't think anything different happened on the Canadian side. So maybe Matt can chime in.
Thanks, John, and good to see everybody there. I'm going to limit what I can say. I'm losing my voice today a bit, so, but thanks for that excellent summary. And you know, the wheels are spinning a bit as I watch and learn about that stalking program, and particularly the pit tagging. So I think it makes sense for us to start scanning those fingerlings if we come across them on the Canadian side, right? Potentially there could be some movement. You never know, right?
Yeah, and it would be nice if you could insert some tags too. We'd get more with the mini pit tags. You could be 10-year juveniles.
Exactly, yeah. And just to update the group, our survey program effort started in 2015, so not nearly as long as what John's been doing. It's only 7 years, and we've been expanding our coverage of the nearshore waters of the Canadian portion of the St. Lawrence River. Last year we were all the way in Prince Edward County, and we go all the way to Montreal as well. It covers a number of different habitat types, including musky sites, which align really nicely with some other species of risk that we're targeting as well. And we've only caught, out of about 150,000 fish, we've only found, I think it's about 20 young of the year muskie at 14 sites. So they remain extremely rare, just confirming the trends that John's been reporting there about very low catch rates. And that's also a significant effort, right? We're doing between 50 and some years over 100 sites in the river, but only find 1 or 2 per year is quite surprising. It definitely remains one of the rarest fish we still come across. And we are planning to continue that survey for the next couple of years as well, moving forward. So just complementing John's work as well, and hopefully filling some of those gaps in on the Canadian side.
Great. Thank you, Matt.
Matt, just a quick question. I'm curious because we know the results from the previous years, but how many did you find last year?
Last year we only found 1 out of 24,000-ish fish documented.
Okay.
And that was revisiting a number of sites that had yielded young of the year muskie in previous years. So even revisiting some of these nursery sites, we've only had 3 sites that have yielded consistent YOI muskie over multiple years. Otherwise, out of the 14, most of them are kind of one-off chance encounters with one. So yeah, it's a very, even last year, they were very rare still. It'll be interesting to see if we see an uptick in the Thousand Islands at all with some of the stalking going on there, though. That's really great.
Hello, John. I'm Alex from the GAN chapter. I just have a quick question that you actually did speak to, but I'm just looking maybe for a bit more clarification. When you say kind of exploratory sites, are you saying brand new sites in different habitat areas, or just habitat areas that traditionally didn't have any spawning in the past and they kind of go through cyclical time periods? And the only reason I ask is obviously with these stalking muskies, they don't necessarily have the nest site fidelity that natural populations have. So I'm just wondering if the exploratory nature of it is accounting for some of the variation where these young of the year may end up because they don't have kind of some of that nest site fidelity. Does that make sense?
Yeah, thanks, Alex. I really appreciate that question. That's a great question. And there's a couple elements to it. So it's specific to the exploratory. So we kind of do two types of exploratory sampling. So the index sites we stick with. And then the exploratory sites come from a list of known musky either spawning or nursery areas that have been identified. So if you remember that map with all the dots, we have something like 120 known spawning and nursery sites. So they represent those. But we also continue to look for sites that haven't been identified, and we actually find them periodically. So that's been a good news thing. We recently found a new site in Lake St. Lawrence not in 2020, found a new site. So it's great to find a site because it's a discovery of another spawning area, and we can try to protect it. In terms of like the question about the reproductive homing, I'm glad you brought that up. So one reason that we're excited about this fry success, we kind of feel that when you use the mechanism of reproductive homing in Muskellunge is not fully understood. The Muskellunge that we catch in our spring spawning index have all been tagged. We've used a variety of tags over the years, and now we've gone entirely to pit tags. But the data has shown us almost invariably that the spawners that we tag are recaptured at the same spawning location. So we call that spawning site fidelity. So we know that they're using, and radiotelemetry and studies have backed this up, that they pick a spawning area and they go back to it year after year. So, and I mentioned pick a spawning area. So the question remains is whether that is evidence of reproductive homing. Were they born there? That is what is not fully understood. And we think that this study may yield valuable information into that process, because if we're tagging these early life stages and they develop a fidelity to that site, it shows that their life history was linked to a specific bay, and then if they go back to that bay to complete their life cycle, it lends credence to the reproductive homing. We could get into the mechanisms of homing and how fish do that. A lot more work's been done in the salmonids, but we kind of feel that when you use an early life stage like it just makes intuitive sense that it would be more likely that they would establish that connection relative to larger fingerlings. So that's one of the advantages of using fry. The cost, and then you get a very wild-like product, that hasn't been handled or influenced that long. So it's linkages to that site or something we're interested in. But also, these fingerlings, we try to get them into the system so they can continue to grow within the nursery habitat for a period of time. and then leave the nursery habitat. And those fish have the tags. You know, the disadvantage is that the fry don't have the tags. We are putting tags in fry that we're capturing later during seining. So I think we captured 168 this year during seining, which was just phenomenal when you compare that to what Matt just mentioned, the rarity. You know, so the fry stocking program led us to be able to capture 168 young muskellunge in our survey, which is quite remarkable. But hopefully that sort of answers your question.
Thanks, John. We've got a question from Rob Myers, who's got his hand up. Thanks, Rob.
Hey, Dr. Farrell, good to see you, and everybody else. Thanks for putting this on. I didn't know if Dr. Farrell, if you were waiting on that guy for an answer on his response. We just asked that question if he was going to respond or not.
Yeah, sorry, my apologies. I got accidentally bounced from the call, so I missed. I'm sure it's a great answer, but
Oh, you missed it.
yeah.
Yeah, sorry about that. It's
No.
recorded. I could do it again, but you know, in a nutshell.
I'll have a look back and go from there, but I appreciate it. Thank you.
Yeah, no
Sure.
problem.
Okay.
Yeah, hey,
Thanks,
Dr. Farrell, just good to see you again. Two-part question. So you mentioned the forage-based monitoring program and kind of that evolving or starting up. So I just wondered, is the angler diary connected to the forage-based monitoring? You know, many of us are probably multi-species anglers, you know, and I don't know if there was an opportunity within the angler diary program to report on, you know, other forage species or whatever that we may encounter. And then, you know, second part question is, if that's not part of the, how you see that program or the monitoring system going already, I guess, do you have more on how you see the forage-based monitoring program evolving?
Yeah, thank you for that, Rob. That's a great question. So as of now, the forage-based monitoring is limited to the nursery habitat. So, you know, we're looking at the small fishes that the young muskellunge depend on and feed on. And, you know, we have a lot of data on that, and we need to mine it. So we started monitoring forage base back in 97, I believe. So we have like an incredible data set on forage. There's a couple studies I did with my former PhD student, Kevin Kapusinski, who's also a pretty well-known muskie scientist. We have a paper out I'd be happy to share looking at prey utilization. But you know, I really like your idea of the angler diary program starting to help us understand forage, because, you know, I know that the forage dynamics and our connectivity with Lake Ontario really influences the forage base of muskellunge, and especially in the fall fishery. And I do get, when I talk to anglers, a lot of information about, you know, they're setting up on schools of alewives, or fish are moving shallow because they're exploiting predators that are, you know, feeding on, you know, forage. So it's, you know, something that we could probably encourage for people to include and take notes on, because I know you guys are using a lot of technology when you're fishing, and you see bait schools and, you know, predators assembling on bait schools that kind of create these, you know, dynamic ecosystems almost that move around. So then the muskie kind of take advantage of those foraging opportunities. And we don't know a lot about those processes, but they certainly interface with our catch rates, right? So, you know, the good and bad fishing is very related to how those things set up. And you guys know that stuff better than I do. So we, you know, more information on that would be appreciated and valuable. And maybe we could think about adopting our diary program to get more like ecological notes about what you guys are observing. So, you know, if there was ever a survey that kind of looked at that stuff, that we would have that information. So thanks for that.
So we're just about at the right time to ask the ministry to come on board, and then we can have some time for further discussions to cross over, I think, John, if you can hang in there with us, because I think there may be some questions about the planning process and updating the strategy and other kinds of things, but it would be great to hear from Colin right now. So Colin, are you there and just about ready to go?
I am. Thank you very much. I'll
Great,
start sharing my screen.
Well, Colin's getting set up there as well. If you've got a question, you can always throw it in the chat area. John, you might be able to tackle a few questions while Colin's presenting through chat.
Yeah, and JP's out there as well. So if JP want to do work on some of those, that'd be great.
Can I just ask which screen are folks seeing right now? Is this full screen or?
It's not yet full screen. We see also the next slide as well, Colin. There you go.
How about that? Okay.
Yep.
And I may just turn off my camera, or once I'm done my introduction here, I may just turn my camera off so I don't suffer from bandwidth here. So first of all, thank you very much for the invitation to speak with you today. I really appreciate it. It's been great so far. Great to get engaged with the Muskellunge community. I learn a lot every time I participate in these things. So my name is Colin Lake. I'm a biologist with the Ministry of Northern Development Mines, Natural Resource and Forestry. And I work at the Lake Ontario Management Unit, specifically at the Glenora Fishery Station. I've been there since 2004, so most of my career. Prior to that, I worked in Northern British Columbia, Northwest Territories, several places in Northeastern Ontario, all fisheries and fish habitat related positions. Once I arrived at Glenora, I was pretty sure I'd found home. It's an amazing place to work. Lots of great colleagues, both in the building as well as colleagues around the lake. So again, really appreciate you having me here today. And I am just going to, if I can, turn my camera off. Perhaps not. Okay, we'll risk it. So I'm here today to speak to you about the upcoming Spring Muskellunge Index Netting Program. A little bit of background about where I work and how we manage Lake Ontario fisheries. So there's a photo there on the left of the Glenora Fishery Station with our ship, the Ontario Explorer, moored. It's a big area of undertaking we have. So our area actually runs from basically the foot of Niagara Falls right down to the Quebec border. It's a big area, and functionally we kind of split it up into three main areas. And that's because of the regional distinct fisheries, and that's a combination of the biological aspects of the fish community as well as the social factors. So broadly speaking, in the West End, the deep cold water trout and salmon fisheries are very important. In sort of the eastern part of Lake Ontario, you start to transition into very important walleye, bass, and lake trout fisheries. And then, of course, the St. Lawrence River with walleye, yellow perch, and obviously muskellunge. You can see the star there I've added. That indicates where our office is located. We're right on the lower part of the Bay of Quinte in Prince Edward County. If you've ever taken the Glenora Ferry across the Bay of Quinte, you will have seen our office there. So because the area is so large and we have these distinct regional fisheries, we manage it accordingly. So Lake Ontario and the St. Lawrence River, as I'm sure you all know, makes up Fisheries Management Zone 20. Our council was established in 2008, which is pretty amazing. It's been around a long time. It's made up of two subcommittees, the East and the West. And that basically is just so we can manage it a little better and reflects the fact that the fisheries are so different. Members are always welcome to join in meetings on the other side. It's not exclusive by any means, but it allows us to focus our meetings a little more either in the east or the west. Long list of participants. We've got a really diverse group of folks represented on the council. There's non-governmental agencies like OFAH, Trout Unlimited. We have some environmental interests at the table. A lot of angling clubs, not surprisingly. The Commercial Fish Association is represented, and we have academia, notably Queen's University and Bruce Tufts. And I'd be remiss if I didn't point out, of course, Muskies Canada. And you folks have been very well represented at the table throughout. You've got a lot of good members that really bring a lot of experience to the table. So you're well represented at the Zone Council. Just one more slide to show kind of the diverse range sort of spatially of the work we would do in sort of a typical year. So this talk is heavily focused on Muskellunge, obviously, but we have a lot of other things on the go. We run well over 20, 25 programs a year. Some of them are offshore. You can see our trawling offshore for alewife, lots of nearshore work, fish stalking, you name it. We're very busy end to end, and also collaborating with our partners on the other side of the border, of course, too. So I just pulled one project out that's relevant to this morning's conversation. So I don't want to leave you with the impression that we've never done anything in the St. Lawrence. In fact, we've had a presence in the St. Lawrence for a long time. So we've conducted a fish community index using gill nets since 1987. And this may speak to one of the questions we just had about sort of prey fish and other forage fish and other species. So we've got an eye on the broader fish community through this program. Starting in '87, we used to alternate this program between the Thousand Islands and then Lake St. Francis further east. So we'd switch back and forth and do one area one year and the other area the next year. Starting in 2019, we've actually changed, so we do both sites every year. It does mean that we have fewer nets. We can't do everything, but this gives us a little more resolution year to year in any changes. So we have 25 net sets in the Thousand Islands, both deep sets and shallow. So we really try and catch as broad a range of the fish community as we can. Typically large-bodied fish. So programs, the same program that goes on really fills in kind of a valuable gap that we don't get. This program is more or less targeted at large-bodied fish. I should point out, it's not designed to capture muskellunge. We don't want to catch muskellunge in this program. Gill nets are typically lethal, and we do not catch very many muskellunge, very few. We haven't caught any for, I forget, a number of years, but we've never caught many in this program, and that's a good thing. It's not meant to count them. But it does give us that important information on the broader fish community. At the end of my talk, I share a link that's to our annual reports, and you can go on there. We report every year. It's a very comprehensive document. It's sometimes nearly 200 pages, and it's got details on this program and all the other ones that we run. So a little bit of history of some of the work we have done at Glenora. This isn't our first entry into muskellunge management and research. We do have a long history, and you can see some of the papers there. I've just shared kind of a representative bunch of them. And Dr. Farrell has mentioned, obviously, Dr. Castleman, who was the research scientist at Glenora for many years. Prominent scientist in the field of muskellunge work. Published widely, many of them co-authored with Dr. Farrell. We are a member of the Socket Working Group. Really good collaboration, and I hope that gets across to you today how tightly we're aligned and how well we're working with New York State DEC, as well as Dr. Farrell and Tibbs, as well as other academics. The Angler Log Program, and you can see a paper there at the bottom, Dan Talon authored. So it's not just biologists at Glenora, it's also biologists throughout the ministry that are involved heavily in Muskellunge. Of course, we played a big role in the VHS response and the subsequent studies in 2005. As I mentioned at the intro there, I started my career at Glenora in 2004. So 2004 and 5 was my first 2 years at Glenora, and it was Dr. Castleman's last 2 years of his career before he retired. So it was an exciting time to be working with Muskellunge, troubling to be certain. But I learned an awful lot. I spent a lot of 2005 that summer driving up and down the river, picking up Muskellunge. The photo that John showed of all the dead muskies on the tarp, I remember that clearly. You know, the smell of dead fish doesn't typically bother me, not in this line of work, but all those dead Muskellunge really took it to another level. It was something else. So learned a lot. Also in 2005, we restarted the juvenile seining, the Muskellunge work, and it really is important to note that Muskies Canada played a huge role in that. Jim Hutchings got a hold of me and said it'd be nice, you know, with all this going on, we should really restart this program. As Dr. Farrell said, it had been running since I think the late 80s by Ann Bendick. And so I took the data that we had on hand, pulled together a protocol, did some number crunching, made some maps, and Muskies Canada really took off and you know, hit the ground running and have continued to do great work, along with Thousand Islands National Park and Matt Windle and the River Institute. So on to our upcoming survey that they're really excited about. So the type of gear we use is we're going to be using 6-foot trap nets, which match very closely with the gear that John and his team use. We'll also be using hoop nets or fike nets. I don't have a picture, but we'll be using these live capture gears during the month of May. And of course, Muskellunge are the target species. However, we will be taking measurements from all of the esocas we catch, and other species we will be counting and releasing. So we will get a sense of the fish community at this time and place. And I should mention that we've really been given a huge head start, not only sharing information with John and his team, but as well as colleagues in my ministry on Lake Erie, Lake St. Clair, and Lake Huron. And I should note that every time I reached out to my colleagues in the ministry and did a bit of name dropping of the folks I was working with in Muskies Canada, they said, "Oh yeah, we work with them all the time." So it's not a new connection. You folks are very well connected and you know, highly thought of. So I'm really excited to participate myself. Another thing I'd like to stress is that our program will be complementary to the existing long-term program run by New York State and Tibbs. And this is just sort of good, sound science. So we want to make sure that the methods we use, the gear we use, the data we collect is able to mesh with that existing data set and make the overall amount of knowledge greater. So I've been working on a protocol. I've taken an awful lot from John's and then a few other things and mixed it in, but the plan is that it will mesh very well. I mean, in fact, if it wasn't for COVID and all those issues, we'd probably be in each other's vessels, learning from one another. It's probably not in the cards this year, but maybe in the future. So as I say, this tight coordination is really important so that our data can be combined with this really impressive long-term data set. A little more about the survey, the upcoming survey. As I mentioned, we're using trap nets and fike nets. Important to note that it's live capture gear. Despite the fact that we haven't run this particular program before, the technicians I work with at Glenora are absolute pros, and they have, in some cases, decades of experience doing trap netting. We do a lot of trap netting, just not in the spring. Typically, our programs are in the fall. So we are far from being rookies. It's just the target species that's new for us. So I'm pretty optimistic we're going to get up and running without too many hiccups here. We're going to use roughly 24-hour sets. So part of the reason for this, obviously, we don't want to be part of the problem here. We don't want to kill any fish. We want to ensure that fish are still robust and swim away happy and healthy. Again, it's live capture gear. We aren't going to keep any fish other than chain pickerel, and I just have a personal interest in that species. They're relatively new arrivals in Ontario waters, so I have some questions I'd like to answer about those. So if we encounter them, we're going to keep a few chain pickerel. And we also hope to get some good information on northern pike through that index gill netting program that I mentioned. The long-term data set is troubling for pike, as it is in New York waters, as it is in Quebec waters as well. We do collaborate quite closely with Quebec, and northern pike numbers, the trends are troubling. So we're going to use this opportunity to get a little bit of information of pike at spawning time. So it's a real value added feature to this program, which is great. Again, identical to John's program, we will be tagging, measuring, and taking a small genetic sample from Muskellunge. And that diagram there is pulled right from the protocol that I've written. So it, it matches very closely or exactly with John's. And in fact, we had a quick meeting yesterday afternoon, and I think we'll probably have a couple more before field season to make sure we've got all the questions answered on our side. So where we are right now, we're still in the planning stages. We're pretty much there, but there is a lot of work to do in the protocol and sort of behind the scenes and how we manage the data internally. And there's a bunch of stuff that doesn't make for a very interesting talk, but there's a lot of work to get a new program off the ground. But we're almost there. And believe me, there's lots of excitement. We're very much looking forward to this. We will be selecting netting sites using information from that the juvenile survey, the seining sites where we know there have been YOYs produced. So we're not starting from a blank page, as well as advice we're getting from John and others. We'll also be making observations in the field. If any of the folks on the call today, if you've got thoughts on some spots where on the Canadian side where you think you've seen sort of spawning fish in the past, you can drop me a line, and my email is going to be on the last slide here. And I'd love to hear about it. Really respect the amount of time you folks spend on the water, and that information is really valuable. We will learn an awful lot during this first season, and we'll apply these lessons as we move forward. It would be great if we could establish some index sites similar to John's approach, and then have some sites, some effort where we're searching for new sites. But obviously, because this is the first year, it's all going to be new. We're going to make our best educated guesses as to where we're setting, but we are going to learn an awful lot this year. And I hope in future years I can come back and tell you about the great success we have. So I think that's all I had. I mentioned there, that's my email address. And then our annual reports are at that link there. I think there's over 20 years of reports there, and lots of information there that you might find interesting. So feel free to reach out. Always happy to chat. And again, thank you very much for the invitation to speak today.
That's great. Thank you, Colin. It's great to be able to have the Lake Ontario Management Unit here, presenting to us, and as part of that, are there some questions from the community for Colin or for the ministry about muskies? Okay, so there's a question here. Matt Lee, are there any muskie stalking discussions on the Canadian side?
Yep, good question. So yes is the answer. We're very interested in the program that John is running. I don't have a decision or an announcement here, but what I would say is the work that John is doing is exactly what we would do to answer this question. So it's amazing that he's doing the work. We're very closely linked to it and interested. You know, there are some challenges, legal loopholes or hoops to jump through in terms of importing fish and all that sort of stuff. So it's not a matter of just getting a green light and doing it. You know, we're going to take the information that John gains and advice that he provides and certainly talk about it. We do stock, not Muskellunge, but we do stock about 2 million fish a year of various species into Lake Ontario. And actually, I'm the stocking coordinator, another hat I wear. So, you know, we're very familiar with the fish culture system and the hatchery system. So, you know, we're interested in asking lots of questions.
Excellent, thank you. I wonder if we can ask, John alluded to this earlier a little bit about the Muskellunge strategy, and it's kind of being updated, and we've heard a little bit about the ESOSID working group. I wonder if John and Colin, if you can kind of talk about the planning process and kind of where we might be in that and what we might see looking forward.
Sure. So we just began initial discussions, and you know, it's going to be a process. You know, we feel like the plan is functioning at this time. You know, it certainly needs to be updated because there's been a lot of science that's occurred, and then these management issues that have arisen. You know, so Colin and I have had a couple of discussions, but you know, we're kind of launching this effort that likely would have been an aspect in the plan. So we're kind of you know, a little cart for the horse. You know, I think the plan will be catching up the, you know, incredible management activities and cooperations going on. So we didn't want to just drop everything and focus on a planning document when we have, you know, a number of major initiatives ongoing, and you know, we're going to do due diligence and, you know, have a good process. And I think, Colin, last we left it off is we're going to have some meetings this summer, you know, when things slow down a little bit, and start to sketch out this process. So we're at the initial stages. Does that sound accurate, Colin?
Yep, no, that's fair. And we do have plenty of other stuff on the go as well. With our agency, we are also in the early stages of rewriting the St. Lawrence River fish community objectives. So they're obviously tied together and linked. We just have a lot of stuff on the go right now, not the least of which is getting this program off the ground and a field program run. So lots going on and lots of good collaboration happening. So not a real rigid timeline yet, John. I think that's fair, but
okay, thank you. We have a couple of other questions here too. Rob reports that Joe has a question. And Colin, while you're here, could you put your email in the chat room? We'll send it out also afterwards when we kind of wrap this up, just so that people know how to be able to reach out to you, Colin. Thank you for that. So Joe, I'm not sure the Joe that's being referred to, but Rob reports that Joe has a question.
It's Joe Enright, Peter.
Thanks. Hi, Joe.
This is for Colin. If they decide to bring in other muskie into the St. Lawrence, which stock would they be considering?
Yeah, so it's a good question, and I'm aware of a few papers that some of John's past students have written on this. You know, again, we're early stages and learning lots from what John's doing, and of course, what John is doing is collecting gametes from the population right here. So, you know, that's something we would want to consider is whether it's best to simply collect those gametes from the population itself and not bring in other fish. But that, you know, that would all be part of the conversation. That's down the road a little bit, but it's a good thought.
Thank you.
There's another question from Robert V that has to do with the St. Lawrence strain versus new strains. So I think, Colin, you just perhaps answered that one for us. Now there's Jim Wilson has one that said, I'd like to see specific stocking 40-acre fish seem to be different from Mallory Town fish. Any comments on you know, that variation that you might see in different parts of the river?
Yeah, sure. That's something, you know, we notice in our sampling that there's kind of a group of different groups of like color and pattern morphs, and I'm sure you guys recognize that as well. The fidelity of those two specific spawning habitats is not well understood, and I think it's something that we hope to be able to analyze. So the earlier studies were done. I don't want to get too technical, but there's a group of genetic markers. They're called microsatellites, and microsatellites are used in looking at population differentiation and assigning. You can use it to assign like individuals to specific clusters or groups that are differentiating. So it's great for determining you know, genetic differences at the population level. But when you look at like specific like traits that genes are expressed that control like you know, things like patterning and other what we call phenotypes. So the phenotype is what the gene that's expressed, and the genotype is the you know, the mechanism for the coding of the proteins to do the job. And you know, so the resolution of the genetics doesn't really adequately look at those individual traits, but that's something that we may be able to do. You know, we also know that there's from the earlier work, there's a migratory population and a resident population in the river. So when that early telemetry work unequivocally showed like two different groups of muskellunge. One group would leave the river to Eastern Lake Ontario from places like across from Mallory Town. We had fish that went to Amherst Island just in a matter of days post spawning. So there's like different migratory groups, you know. So we're hoping that both the genetics and perhaps some future studies that we hope to do with the new acoustic array in the river, better understanding the biology at that level. So thanks for that question. But we do take another part. I think Colin may have mentioned it. We do a photograph of each individual fish. So if you know, that's another place where anglers could really help if they're photographing their catch, you know, in a good side view of the fish without, you know, all the gymnastics that go with photographing fish. But you guys are really good at it. It's that would be interesting to start to characterize the frequency of these different color morphs, if you will, that exist in the river. So we have some data on that too. So thanks for that.
Great. Thanks, John. Rob McRae has a question. Go ahead, Rob.
Okay, no, I just had to get myself unmuted. And this is basically for Colin and John both. When was the last time we did an active genetic survey on the Muskellunge along the St. Lawrence River and East Basin of Lake Ontario? I have a ballpark, and I know we're about to step into it again. I just want to know the breadth of time that we've put between them.
Yeah, so there's been the recent major studies was the one that I did that you're probably aware of with Kevin Kapusinski. It was part of his graduate work. So I think that was it was published in 2013, I want to say. So I can forward these documents to anybody who's interested in, there's a lot, it's pretty heavy with jargon in the scientific field of genetics. I'm not a geneticist, but you have to pretend to be one in order to understand fish populations. And yeah, that came from the same sites that we've been working in, and it integrated. We had archived samples. So, and now I'm kind of looking for another collaborator at the moment. But I mentioned Louis Bernacci, Lab at Laval University. He's a Canada Research Chair, one of the top labs in the country. And we worked with him and his postdoc to look at the entire system. So we participated in a study that examined the Thousand Islands, but the Thousand Islands were more of an outgroup for a broader study that was in the Quebec waters of the St. Lawrence. But we learned a lot about the influence of stocking, like I mentioned, with integration. And now we have a tremendous Muskellunge tissue archive. So all those fingerlings are fin clipped and preserved if they're above 80 millimeters. And all the adults are fin clipped and all those samples are preserved. And, what I hope to do is we have the parents as well that were used in the broodstock collection. All those are archived. So I have those crosses, and we're hoping to do like a parentage study, kind of understanding how like the different broodstock that we use, their success in helping to reestablish the populations. We're expecting there'll be variation in that. And then another thing we're looking at is, looking at what's called effective population size. So you can use genetic tools to understand like the influence of a single female on a population of young. So, we could start to look at the young of the year and understand how many females contributed to the young of the year that we see. So if it's just a single individual, that's like alarming that all the young would be coming from the same parent. You want to see a diversity of parents supporting a population. So it's a kind of a critical management piece, and having the ministry working and collecting data on the Canadian side where we think there may be even more habitat. That's very hopeful. So it's really exciting that this genetic tissue bank is going to increase, and then we will assign a future postdoc or graduate student to extract DNA from those samples and really look at the system like it's never been looked at before. And that's going to be coming, probably at the, after we continue to build, we're going to get more and more tissue data. The citizen science program is going to contribute tissue, and we're going to have a really good picture of the genetics of the St. Lawrence population.
Okay.
We have. Thank you. We're coming up to the end of our official time together, but this is such an interesting conversation. Maybe if John and Colin, you can hang on for a little bit. We can maybe pursue it a little bit further here. Jim Wilson has another comment to everyone. He says he does appreciate color differences, but his observations are 40-acre and Clayton fish are much more stocky than Mallory Town fish. So in other words, there's some phenotypical differences that are not just color, and perhaps you can comment on that, John, if that might relate to migratory fish as opposed to resident fish.
Yeah, I've always felt, I'm from Oak Point, right across from there, and that's where my family has their camp, and I always felt that the Mallory Town, and I'm not that familiar with it, like Peter and others are, but I think there's a pretty significant population of younger Muskellunge there, that use that area, not only for subadult development, also like these earlier adult age classes. So it seems like they're kind of at higher density. So it might just be a like a regional growth phenomenon and age structure, but that's pretty much pure speculation. But I know if you go downriver to Oak Point and then, Cold Ferry and American Island and places, those traditional musky habitats, which were, once there's still, a lot of fish there, but it was incredible. They get really large individuals around Brockville in that area. Really, really big fish are being caught throughout the system. So I kind of feel like that's a kind of a unique and important place, that grows muskellunge. We see that on the backside of Grindstone too. We also see, probably these subadults are less frequent. We don't see them in our catches because they're out in the main river and deeper habitats that are beyond the limits of sampling, but we do see them more frequently on the backside of Grindstone, these younger muskellunge, and people are catching younger muskellunge, which is great news. So I hope that helps. It's kind of speculative, but different regions of the river kind of serve different purposes for their life history is likely in different habitats.
So can I ask, speaking about younger muskellunge, and you're coming up to potentially age 5 for some of the earliest fry that were released. Should the angler community be watching for and paying particular attention to whether or not we're catching, say sub 36-inch fish, those that might be 5, 6, 7 years of age? What should we be watching for as indicators either of problems or of reassuring news on those younger fish that we seem to have lost a number of young of the year classes of?
Yeah, so there's processes, and Colin is trained and can speak to this as well, but fish populations in general, like if you have really fish populations at risk, you might be missing like age classes and cohorts in your population, and we're not seeing that in the St. Lawrence. That it seems like the age classes are represented in what we see and catch, and that's important, because if in populations that are really, really at risk, you might have missing cohorts and age classes, which would really put species at risk. There's a couple concepts in fisheries, like there's like a density dependent compensation, it's called. So sometimes when fish are really abundant, they'll actually compete for resources. And if you get a year class that's not as well developed, it actually presents an advantage to those fish to be able to exploit prey, maybe have lower mortality, and the performance of the remaining fish can compensate, and they can actually perform pretty well. So there's a compensatory process that can happen. And we don't fully understand a lot of these dynamics. We're working from indices to get accurate aging structures from muskellunge. Unfortunately, you have to use fin spines or Clythra bones, which require the fish to be dead in order to age it, that's unacceptable. I mean, historically, we worked with taxidermists. That's where John Castleman built the Clythra database, and we sent him hundreds of Clythra from taxidermists. Fortunately, the taxidermists aren't as busy stuffing muskellunge as they were historically. So, there's less like age structure information. So it's a bit of a challenge. But that is something to be optimistic about. But we're still very concerned about what you heard from Matt and the density of young coming off the nursery grounds. It's kind of a long process that will erode, it's been eroding and will erode the population. So, there's this concept of shifting baseline. So people begin to accept, what is current is acceptable. But when you put it in the context of what we had in the past, it's just a fraction of the population that we had historically. So, and people are better at catching them than we were back then. So it's, technology helps people find fish, but we're hopeful that this new citizens program will help us better understand the population beyond just an index. So, with the tagging program, there's the possibility that we could get a population estimate, which would be a first time and an incredibly valuable piece of information to know how many fish are really out there. And you do that through mark recapture. So hope that helps.
Thank you, John. We will send out coordinates and information about how people can participate in the citizen science program, as well as the email addresses for Colin and if people want more information or the ability to follow up. And we'll also keep the chapters involved, Muskies Canada, Muskies Inc., so that we can kind of continue this. So we'll do an email follow-up at the end of this. Alex Rowe had another question, or he had his hand up there. Alex, over to you.
I'll just try to be brief. Something that John said just kind of piqued more of my interest and made me think about something. With the aging structures and kind of the need to have them deceased, I'm just curious if all of those collected fish from the VHS virus in years past, other than kind of infectious disease analyzing, did a lot of those fish get used for some of these life history variables or kind of other metrics to identify anything in the population?
John, I'm not sure your microphone is on there.
Yeah, John Castleman really from the Glenora station at that time led the charge and made incredible use out of those dead fish. So it's in Colin's database, and John analyzed that data and looked at the segment of the population that was being most impacted. And we have a paper that's in the book, the Muskie management book, that utilized those issues. He extracted ovaries, other organ tissues, and all the aging structures, and integrated that into the current knowledge. And tremendous use was made of I think it was roughly 102 Muskellunge that John used in his database. So it really did help with the understanding. And I think in a nutshell, we saw that unfortunately that the disease selected for these large and older female Muskellunge. So that seems to be the key variable that makes us think that some of the population decline and the loss of recruitment was related to that outbreak, and it has a legacy with it. So there's you guys are still catching a lot of big fish, which is great news. I think muskie are growing faster. We know the river's warming. Got a lot of data. It's incredible, the warming in the river, especially in fall and winter. So, those fish that are age 5, should be entering the fishery this fall.
Colin, did you want to comment? Go ahead.
Yeah, I can just add a little bit to that. John's absolutely right.
Oh, we just lost you, Colin.
Sorry about that. Yeah, I was just going to add, we used everything from those fish. I remember doing the necropsies with John Castleman, and when VHS first started, of course, you recall, we didn't know that's what was killing these fish. And the first couple of phone calls I got was saying, I have this large Muskellunge that washed up to my dock. Do you want it? And we said, absolutely, that would be great to have. I didn't know that my phone would ring off the hook for the next 6 weeks or 8 weeks, but the point is we don't catch a lot of Muskellunge in our surveys. We don't want to, like I said. So to begin with, it was a real resource. And then once we had all those fish at the station, we still didn't know what was killing them. So we looked at everything. I mean, there was almost nothing left over when we were done. So it was very thorough. And I think a lot of it's detailed in that paper, John.
Yeah, thanks for the comments on that. I'll just have to familiarize myself with some of that. I wasn't aware the breadth of kind of data that was taken from that. Mhm.
Well, we're just a little bit over our time. Bill Barber's got a question here in terms of, with his observations of spawning pairs, he's noticed goby predation on the eggs has been devastating. Has there been any thought on habitat restoration around Wolf Island or the Kingston area?
Yeah, thanks for that, Bill. We actually did a study, and we're proposing another study on goby egg predation. So one of my graduate students set up experimental treatments in our lab with differing musky spawning substrates, and then also some treatments that had very little habitat, like sand and bare bottom. And then we had algae, we had aquatic plants, and then cobble that have interstitial spaces that protect eggs. And we seeded them with treatments. One treatment was with northern pike eggs, and a second treatment was with Muskellunge eggs. And then we put a single goby, and this was replicated over and over and over. I think it was 6 or 8 times it was replicated each treatment. And we put a goby in there and let it stay for 1 day. And we put 30 eggs in each treatment, and like the bare and unprotected substrates, there was like 100% predation on the eggs. And then the more complex habitats had, they did protect the eggs somewhat, but we still saw 50% of the Muskellunge eggs that were consumed by gobies, even in complex habitats where the eggs kind of fit in, and so they were able to sniff them out. People have studied like bass, worried about nesting and stuff like that. And we were curious to see if the gobies could find eggs that were broadcast over complex substrates, and sure enough, they could. It was very concerning that a single goby could find half the eggs in 24 hours in a pretty large treatment. So we're thinking of doing this now in a field situation. Usually you start with an experiment, and we're going to try to do a field study to understand whether this is another really significant stressor. Even if we restore a population, if gobies are highly abundant in eating the musky eggs where they get to hatch, that would be very, very concerning. So, not only do the gobies harbor the virus, but they serve as significant egg predators. So that's something that we're looking at. But maintaining like really quality habitat is just, that's our foundation. So, we really need to protect our habitat, and it's, like I said, it's changing, and people are developing habitat. So we need to really take care of it. Thanks for that, Bill.
Great. Well, I think that's pretty much covered the range of questions that we've got. John and Colin would really like to thank you on behalf of the Muskie community. It's great to be able to get an update on, the situation, on what's being worked on, the excellent research and planning work and assessment work that we're looking at coming into the next year. And, the whole appeal to the Muskie community to be citizen scientists falls quite naturally with us, especially the way we report for the Lunge Log for Muskies Inc. and the Angler Diary for your group, and for Muskies Canada's Muskellunge log program. So we'll make those coordinates available and be able to keep in touch. On behalf of Muskies Canada and the Gananoque chapter and all of us in the Muskie community, thank you very much for what you do, and thanks for sharing that with us this morning.
Thank you.
Thank you. thanks for putting this together, Peter. And we'll see you guys on the river, hopefully.
You bet.
It's
Thank
okay,
you very
John.
much.
We'll get you out there.
Thanks,
Cheers.
everybody.
Thank you.
Take
Thanks,
care.
everybody. See you later.
Chat recording.